Bismillahirrahmanirrahim. 5090 O-Level Biology, New Syllabus 2023, Chapter 7, Video 1. Now let's look at a root hair. And what is a root hair? You can see this diagram here. We have this diagram here where the root hair is, and then we have a large diagram of it. So it's a epidermal cell. It's got a cell wall. It's got cytoplasm. It's got a vacuole. It's got a cell membrane, nucleus. And this is a fully grown root hair, while these are the newly growing root hairs. So this is the one which has fully developed, and these are the ones which are all developing. So they will keep on developing as the root system increases in size and in branching networks. Now root hair cells are very important to the plant because roots do more than just anchor the plant in the ground. They take up water and mineral salts from the soil. The surface of the cells are specialized root hair cells with a long thin extension. You can see this long thin extension. This one again has a long thin extension that reach into the surrounding soil. The root hairs provide a large surface area for substances to enter the root. Then the adaptations which they have, root hair increases surface area. Then they have thin cell walls because the cellulose cell wall is thin. Why? Because so that all the water can easily enter by osmosis and the ions can enter through the cell membrane. So large permanent vacuole contains sugar, so there's low water potential. This is the sign for water potential. And the reason they have a lower water potential is because then water can enter through the root hair cell. Water can enter from a higher water potential to a lower water potential. Then these are positioned very close to the xylem, and they have a lot of many mitochondria for active transport because the ion uptake is by active transport. The water enters by osmosis. Water will enter by osmosis, but the ions will enter by active transport. This is how the root hair cells look like in real life, and just to make you understand the enormity of the situation and the large surface area which they are exposing. So now this is a diagram which is of course not a very good diagram because my drawing is not very good. But a normal root hair cell has a cell wall, which is thinner, of course, because so that it allows all the substances to enter. So we've got the soil particles and we've got the water in the soil, and so the water is going to enter by osmosis. Why is it going to enter by osmosis? It's going to cross the partially permeable membrane, the cell membrane, which is partially permeable, and the tonoplast, which is partially permeable, which is the vacuole membrane. So water enters by osmosis because it will be a higher water potential outside. So there's going to be a higher water potential here in the soil. And inside, because the vacuole and the cytoplasm have solutes in them, so it will be a lower water potential. So water will enter from a higher water potential to a lower water potential. And this is what I've shown you by this arrow. Then we come on to the next important thing is the ions. So the ions which are present here, these are, I'm making these in red. It could be nitrate ions, could be magnesium ions, could be potassium ions. Now the ions can only enter by active transport. Why? Because there are ions inside this as well. And there are usually more ions inside than there are outside. So more ions here, but less ions outside. So now how will they enter? They have to be entered by active transport. And for active transport, you need a channel protein in the cell membrane. Not in the cell wall. You need channel proteins in the cell membrane. And the gray line is the cell membrane, which I've written it here. You can see this. I've written it in the same color. So there will be channel proteins for active transport. And these channel proteins will allow the nitrate ions and the phosphate ions and the magnesium ions to enter through these channel proteins. And of course, the number of channel proteins is that limiting factor here. And you have one MCQ on that, very important topic. But plus another thing, for active transport, you need energy. Now, how is the energy going to be provided? Energy is going to be provided. There will be lots of mitochondria here. And the mitochondria is going to get the oxygen from the air spaces in the soil. And aerobic respiration is going to take. You know oxygen, there are going to be air spaces here in the soil. because if the soil is compact, then the plant dies. So air spaces in the soil and oxygen is going to enter through through the cell wall and the cell membrane. Cell wall, of course, is fully permeable, and aerobic respiration will take place and energy will be released, which is needed for active transport. So ions will be taken up by active transport. Now this is the function and the structure of the root hair cell, how it is adapted for the function that it needs to perform. Now the next topic which we have to understand is how is it going to travel up the plant? How can water travel up the plant? There has to be some suction force which is being exerted, and that suction force is water evaporates from the stomata. So water around the spongy mesophyll cells was in a liquid state, then it's going to become a vapor state, and then it's going to diffuse out. So water vapor is diffusing out of the stomata. So water evaporates and then comes out of the stomata. Now this is going to produce a suction force here. So you have to start where the beginning is here, the end is here actually. So it's the water evaporating and then the water moving out of the stomata which causes a suction force, and that is going to cause the water to be pulled up the xylem. So let's look at it from both sides. First of all, water moves into the root, then it is going to be moving through the cells of the cortex of the root. These are called the cells of the cortex, and then this is going to enter the xylem vessel in the stem, and then the water is going to be pulled up with the xylem vessel, and then the water enters the cells in the leaf, and then it is going to evaporate, and then water is going to move out of the stomata. But basically this is the beginning, this is the suction force, the water evaporating and the water vapors moving out of the stomata. Now another brief overview, I like this as well. Water uptake from the soil, additional water enters through the roots. Then there's cohesion and adhesion in the xylem. Cohesion is the fact that the water molecules stick to each other, and adhesion is the fact that the water molecules stick to the wall of the xylem vessels. So the cohesion and adhesion causes the water to rise up. So water column rises by cohesion and adhesion, and then water evaporates from the leaf surface. So this of course results in the transpiration, which is a force that pulls water through the xylem. Now, why am I giving you all these different explanations is because I want you to develop the skill of writing the answer. So these wordings and this biological grammar or the biological language is very important that you understand it and you are able to write this. So the first thing which I want to read to you is this part, which water moves from the soil to the root here, from the soil to the root here, then the cortex of the root. This is the cortex of the root. You've got to understand is this area is the cortex of the root. And then xylem in the root, and then xylem in the stem, and then xylem in the leaf, and then they leave the mesophyll, then air space, and eventually through the stoma to the atmosphere. Why? Because the atmosphere has a low water potential. There is a water potential gradient from the xylem in the root to the atmosphere. Evaporation of water from the mesophyll causes water molecules to be pulled up the xylem. That is called the transpiration pull. And cohesion between water molecules and adhesion between water molecules and the wall of the xylem vessel maintains a continuous column of water. So this understanding you must have is why water is being pulled up, is why is the water being pulled up in the stem. There is a reason why, because water is moving, water vapors are moving out of the leaf. Another very good diagram to show you this, which I want you to see this. You see, this is the root hair cell, and these are the cortex cells. These are all the cortex cells from here to here. So first the water is going to enter the root hair cell, then it's going to go from cell to cell in the cortex, and then finally it is going to reach the xylem vessel. And then of course is the fact that it's going to be pulled up. So you can see the cohesion bonding of the hydrogen molecules, you can see the adhesion bonding of the hydrogen molecules, you can see the transpiration flow, how it is going to be pulled up, and the fact that there's a negative hydrostatic pressure in the leaves. And of course, this is going to help in pulling it up from the root hairs to up into the leaf and out of the leaf. Now, in 7.2, there's the first point is describe transpiration as a loss of water vapor from the leaves. So the first thing which you've got to understand is that the water molecule is going to move out of the xylem, but this is not osmosis. Why? Because xylem does not have a cell membrane. We will be discussing this in the later part of the video. And then the water enters these cells, water will enter this cell, water will enter different cells. And then the water moves out of the cell and comes to the surface of the cell. It's a liquid, it's a film. The water is in a liquid state. Now here the water has to evaporate and become a vapor. So these are the crosses which I'm making in the air spaces. These are the water vapors. Now these water vapors are going to diffuse out. So basically on the surface of the spongy mesophyll cells, there is a film of liquid. So it's still in a liquid state. But then as soon as it converts into a gaseous state, then we don't call it water, we call it water vapor. And then these water vapors in the air spaces are going to diffuse out. Water vapors are going to diffuse out. There's not osmosis, there's no partially permeable membrane involved. So describe transpiration as the loss of water vapor. Vapor is very important that you write this. If you write water, I can't give you any marks. So water vapor from the leaves. So basically water comes out of the xylem. That is not osmosis again because there's no cell membrane. Then water moves into the cells of the palisade and the spongy. Then they come to the surface of the cell. And then from the surface of the cell, they convert from a liquid state to a gaseous state. And that would be classified as evaporation. And then the water vapors will diffuse out of the leaf through the stomata into the air. Basically, we have got to understand where the xylem and phloem are located in three organs: leaf, stem, and root. Now, you see the section here has been taken from here. Now, the xylem is in the center, and this is cross star shaped structure is the xylem, and the phloem is on the sides of the xylem. So you can see here the phloem is here. This is the phloem, this is the phloem, this is the phloem, and this is the phloem. So the xylem and the phloem in the root is right in the center, and the main vascular bundle is right in the center of it here, because the root has to burrow into the soil, so it has to have strength. It has to have a central core which is strong. And this is called the epidermis, which is the outermost layer, then the cortex, the root hair, and the rest I won't say because that's not in the O levels. Now, when we take a section from the stem, now here is very different. We have vascular bundles. So we have a vascular bundle here, here. Now these are all at the periphery because they're going to give strength to the stem, because the stem has to withstand wind. The root does not have to stand that. So if there is a windstorm, so the stem has to be strong. Now that has the vascular bundles are all on the sides. So in the stem, this is the stem you have to remember. So this diagram is showing you the distribution of the xylem and the phloem, and the xylem is on the inside and the phloem is on the outside. So this is the xylem inside and this is the phloem on the outside. So the outside has the phloem and then the xylem is on the inside. If I give it another color, so this would be the xylem on the inside. And that's why you can see the aphids can suck fluid from the phloem, which is a sugary fluid because it got sucrose and amino acids. That's what Coca-Cola has, sucrose and amino acids as well. Then coming to the leaf. Now when we cut a section of the leaf here, you can see this. The leaf has the midrib and it has got the xylem is on the upper side. So the upper epidermis, xylem is in the upper side. That is why I make you remember it like this. Leaf upper xylem. Leaf upper xylem. So the xylem is on the top and the phloem is on this side, so the lower side of it. So this is the phloem and then that is the xylem. So we have to know the distribution of the xylem and the phloem in the leaf, stem, and the root. So this is the main vein, and then you've got the smaller veins on this side. And this would, of course, be the entire leaf, and we've shown you a section of that leaf and taken a section of it here and shown you this. So xylem is on the upper epidermis side, phloem is on the lower epidermis side. In the stem, the vascular bundles are on the periphery. In the root, the vascular bundle is in the center, and there's one big vascular bundle, while in the stem, there are many smaller vascular bundles. The other comparison between xylem and phloem is xylem is one-way flow, this is two-way flow. The minerals and ions, water and minerals, no end walls between cells, and they are stiffened with lignin. They've got lignin deposited in the wall. While here is water and sucrose and amino acids, we call it food, but I don't agree with the word food being used. And the end walls have perforations, and these end walls have perforations, and that's why these are called sieve plates. Sieve is a sieve like you sieve your tea leaves. So these are called sieve plates. Another diagram showing you the distribution of xylem and phloem, the stem and the root. I hope everybody is better clear because some diagrams are not very explanatory. So I like to give you a few more diagrams to be able to understand. You can pause the video here, have a look at it and see if you've understood it. Now, how do xylem vessels develop? That's a very important fact that you must know. It's not probably given in your books, but I like to explain it my way. So once upon a time, there were normal elongated cells joined end to end, which I've just drawn these ones. And they have a nucleus, they have tonoplast, they have everything. And then what happens is, so normal elongated cells joined end to end. So normal elongated cells joined end to end. Now cross walls break. You can see here I've shown you the cross walls break. And the cell contents die. So there's no more any cytoplasm, there is no nuclear membrane, there's no nucleus, everything finishes. And lignin is deposited. Of course, there are different ways lignin is deposited. Sometimes it's deposited in a spiral form, sometimes in other forms. And now what happens is a continuous column forms, and this transports water and ions from the soil to other parts of the plant. But basically, the lignin provides a lot of support because lignin is very hard. Now, in order for these xylem vessels to transport water in very tall trees, as many, many meters high trees, so it has to be a very, it doesn't collapse. It doesn't have to be something which will easily collapse because the force of the water being pulled up is immense. When many leaves develop and the foliage of the tree is huge, then a lot of water vapor evaporates from the leaves. And this, of course, creates a massive suction force. And so this water is pulled up in the xylem vessels. And the strength of the xylem vessel is because of the lignin deposited in the wall of the vessel. But please remember, you say xylem is dead. Now that is not very correct. Why is it not very correct? But anything dead is not hollow. Like a dead bird is not hollow. A dead tree is not hollow. A dead lizard is not hollow. So when you say dead, you mean there are no cell contents. You mean there's nothing in it. There's no cytoplasm. There's no cell membrane. There's no organelles. There's no mitochondria. There's no nucleus. But by saying xylem is dead, that's incorrect biological English. So you can say, yes, it's hollow. You can say cell contents no longer present. You can say continuous columns form, transports water and ions. As you can see here in the syllabus, now this is the point which I've tried to explain to you all is number 8 point, state the function of xylem as transport of water and mineral ions and support. Then relate the structure of xylem vessels to their function limited to thick walls of lignin. Thick walls with lignin, details of lignification are not required. And no cell contents and cells joined end to end with no cross walls to form a long continuous tube. So this is the exact syllabus points which I have tried to explain to you all so that you can understand, because you see whenever the paper is being made by the examiners, they have to tell us, they have to tell the Cambridge examining board is which part of the syllabus are they examining. So if you have gone through the syllabus very properly, then there is no way that you don't understand this entire subject. So following the syllabus and understanding the syllabus is of course one of the key study skills which students must develop. This video will be finished here because I will continue this in the other video. There are a few points left in the syllabus still to be covered, and translocation has needs to be covered, and we have to study what is a potometer, and we have to investigate the rate of transpiration, how it alters with different factors. So thank you for now, and inshallah very soon I'll be putting up the second video which will complete this chapter.